Welcome to Nucleic Acids Part 4. In this tutorial, we'll give a brief introduction to genetic mutations. Let's get started. Alrighty, so what is a genetic mutation? It's any permanent chemical change at one or more nucleotides in the DNA sequence that affects the primary structure of a protein. So we're going to focus on two different types of mutations. There's the substitution mutation, right, where we're going to substitute a nucleotide, like valine for aniline, something like that. The other one is basically where a frame shift, and this is where we delete a nucleotide, right, so it gets lost. The lost nucleotide. Alrighty, so let's see if you can recognize the difference. So here we have the DNA template, and then here we have the messenger RNA strand, and so here is our polypeptide sequence, and now we'll look at A and B. Alright, so we scan for a difference, and as we look across, as we scan, we see matching, voila, and there's a little line there to help us, right? So we notice that everything is the same except that the T has been replaced or substituted, right? Actually it replaces or substitutes for the G. Okay, and so then we note what happens there is that we want alanine and we end up with aspartate. So it's this amino acid right here. So notice with the substitution that everybody else remains the same, and if we look afterwards, the leucine matches. So here there's just one amino acid that has been replaced or substituted. Now this looks very different from the frame shift. So once again we'll focus on the same nucleotide. Here we see that the first place of difference is the G is now an A, but if we continue with the pattern we see A, T, A, T, G. So this is an example where we lost G, right? So that created the frame shift. And this one has more far-reaching implications because not only does it change the codon sequence for the amino acid where the frame shift occurs, but now notice that every amino acid after the frame shift will also be changed. Right, so these are the two types of genetic mutations that you want to be able to recognize. And so remember, sometimes this can get kind of creepy to think about, but remember that these mutations are minimized by the fact that there is more than one codon for most amino acids, and there is editing and double checking that takes place through our biochemistry. So DNA mutations can create defective enzymes, and so examples of mutations that we can live with are like sickle cell anemia, or even people that are lactose intolerant, right? So people that are lactose intolerant don't have the enzyme to break down lactose, right? But that's something that we can either avoid dairy products or take advantage of lactate, that kind of thing. However, some DNA mutations we cannot live with, and basically what happens there is death. And often this death happens before we're even born. And so just to wrap up here, so mutations can come from a variety of factors. We can have a predisposition in our genetics. There are certain chemicals that will trigger mutations, high energy light, UV, X-ray, and gamma rays. So that wraps up our tutorial on genetic mutations. So please take some time now to work a few homework problems to reinforce your understanding.